pull up a chair, take out the oxygen, and join the discussion.
You've just stepped into life unculturable.
The world where conversations grow faster than colonies.
We're your hosts, Sam and Julia, scientists by day,
Podcasters by night,
and professional oxygen avoiders 24/7. Join us in exploring the science of microbes that thrive in the absence of oxygen on Life Unculturable.
Okay.
We
are live and ready to go. Making media would really suck. Is what I'm reading off the script just now.
Well, I mean, I try to keep it real. So,
oh my. Okay.
What's a cold opener? I'm like, hey, Julia, have you checked the gas mix recently?
No.
You haven't? Probably because I stole the last nitrogen tank yesterday.
Man, Sam.
I know those are a hot commodity in the lab these days. And,
well, you can have all the nitrogen.
Subsequently, that's the one that I broke the little plastic piece off of the regulator. Shout out Brian at Koi. I do appreciate you hooking me up with a part. I think Julia and I are going to talk a bit about the Koi chamber today, or a chamber rather. Whether you've got a Koi chamber or you've bought one for another company, those just happen to be the ones that we have access to here at OSU. But that's probably one of the more common pieces of equipment that we actually use when we're dealing with anaerobes. What else do you think is pretty common when we're dealing with anaerobes?
I mean, a gassing station is probably very common.
Yeah, you don't think most labs have a gassing station?
I think most do. Like most anaerobic labs.
Oh, yeah,
should
most
have,
anaerobic
yes.
labs. But just the regular.
Oh, the regular people do not.
Labs.
The regular people do not.
The cool microbiologists.
Yeah. So I think the anaerobic chamber is one that is even, I think, rarer even amongst the anaerobic microbiologists because it's not strictly necessary.
Yeah.
And it's like a big piece of equipment that is quite expensive and tedious to maintain as well.
Yeah.
But the gassing station, I think that is pretty much necessary.
I was going to say, is that the most important then?
I think so, right?
Yeah, like those are probably the big two fixtures in our anaerobic lab life, are the gassing station and the chamber.
If we had to rank all the things we use
in
like most important to least important in anaerobic work, how far up would you rank a crimper?
Okay. So I hear what you're saying, and that's one of those where it's like, what's the greatest invention? And it's like cell phones, but you got to go back to electricity. It's like no cell phones without electricity. And I love the crimper idea, but in our case, you wouldn't really get very far crimping things if you couldn't make them anoxic, which I guess you could with,
it's
like
a hen or the
scrubber
egg problem.
Yeah.
Okay.
But yeah, in general,
gassing station at number
I
one.
think, let's say lab equipment that's larger lab equipment. We'll ignore the Bunsen burners and the syringes and, all of those things.
And we count the gas tanks as consumables, not equipment.
Oh, we do consume them. I mean, how many gas
regulator
tanks?
And all.
Yeah. Oh, I know. As of just a few days ago when I broke that regulator, like there's $1,200 down the drain. But yeah, that's tough. Those gas tanks are, I'd say, a hot commodity, but I mean, that metal's cold. So how many gas tanks do you think we go through in a month?
I think it really depends on what's going on in the lab.
Mm-hmm.
We're going through more gas tanks, or we went through more gas tanks when the anaerobic fungi side of the lab was also in full swing. Currently, it's quieter in the lab for anaerobic fungi because we're doing like paper writing and data analysis and stuff like that. And you should be happy that it's quieter because you have the lab to yourself now.
I mean,
mostly.
Yeah, mostly.
Other than the culture collection, of course.
Yeah, that's true. I can't get away from that.
So anaerobic fungi-wise, I think we go through currently one CO2 gas tank a month, which is not a lot.
Yeah.
We used to go through them in a week, max 2. And then the nitrogen gas tanks, I feel like you go through a lot of them because you like maintain the chamber with it, you do media prep with it. We sometimes snack some of the nitrogen because we do antibiotic preps with it.
Mm-hmm. Yeah. No, that's a great point. And I think I do go through a lot more nitrogen than you all.
Mm-hmm.
probably do because we maintain both the anaerobic chamber as well as the hypoxic chamber. Both of those suck a lot of N2.
But yeah, I think overall we're going through quite some gas tanks, but it really fluctuates depending on how much we do in actual lab work. And also ever since we moved the anaerobic chamber for fungi from nitrogen to CO2, we're not going through that much nitrogen anymore.
Oh, yeah, I didn't think about that.
No. So, yeah. But maybe let's rein it back in because I think we've gone off the rails here a bit because we were talking about common pieces of lab equipment that the anaerobic lab has that aerobic labs might not have.
So the key word here is common.
Potentially.
Okay.
So we've ranked the gas station probably number 1 as most common and most important.
Whoa. I may not rank the gas station as the most important.
Is it now gas station or gassing station?
You know, we're going to go gassing station. But I do agree with you. I think it's more important than the chamber. I just wanted my voice to be heard, is all.
But you just said you wouldn't. What?
I was just being dramatic.
Oh,
I got a gassing station, hands down.
I need more caffeine, apparently.
All right. If you were listening out there, bring Julia a large, giganto cup of coffee, please.
Black.
Yep, and that fancy kind.
The fancy kind?
Yeah, the kind you make in the sand.
Oh,
what's that?
Yes, the Persian coffee.
The extra caffeinated
Turkish coffee or whatever.
That one. Yeah.
Yeah. Love it.
I'd say we'll take 2 of those, but that might get dangerous. So let's just go with one. One of those, please. But jokes and drama aside, what types of... So I guess do we go consumables now instead, or consumables / small lab equipment that you think are pretty common for us to use?
Let's maybe finish off the equipment part.
Okay.
Like large and small lab equipment, and then go into the consumables, I would say.
Okay, let's do it.
Just to keep some structure in there.
Yeah, we probably need structure. By we, I mean me.
Don't we all? But yeah, so gassing station, although gassing stations are not commonly obtainable from just like any company or anything, I don't think there's a company out there that would like make a non-customized gassing station for you. True. Right? You always have to like either build it yourself or you just raw dog it and put some tubes on a gas tank and call it a day.
Yeah. So if there's anybody out there that is in love with making gassing stations and you would like to make us one, go for it.
Yeah.
Because I think every gassing station that I've ever worked on in an anaerobic lab has been homemade in that sense. Mm-hmm. It's like somebody bought these pieces from this big store and then this thing from over here, and then it gets pieced together. Mm-hmm. And then that's just how it is.
Yes.
But we need a streamlined version.
There we go.
All right. That's our Shark Tank pitch. We're going to go on there.
So it's gassing station and autoclaves we need built. Hey. Aerospace engineers, come on.
Leave the autoclaves out of it. We have a brand new one in the lab.
We do, and it works really well.
Somebody hear that knock on wood? It is working fantastically right now.
Yes, it is.
It's been, 2 weeks?
Yeah, maybe a week and a half. Yeah.
Oh, come on.
We will keep you posted on how long it lasts.
Yeah, we'll have like one of those little calendar stickers where it's like so many days since the last workplace accident. It'll be, so many days the autoclave has been working. So we'll just keep adding.
Yes.
Okay.
Oh man, we're going off the rails again. Lab equipment, right. Gassing station, we've dealt with. Yep. We've rambled about it enough, I think. Next one is anaerobic chamber, is something that many labs don't have. The hypoxic chamber is, I think, something that only very, very few labs have, right?
Yeah, I would agree with that. I think there's probably less labs that have the hypoxic capability than like the total anaerobic capability.
Do you want to give us a quick rundown what the difference between the anaerobic chamber is and the hypoxic one? Sure,
I'll give it a go. In general, it's just the utter absence of oxygen in the anaerobic chamber. And then in the hypoxic or the microaerophilic chamber, we've got, some small percentage of oxygen. But it's going to be under what it would be regularly just in the atmosphere that we're enjoying breathing right now. So currently our hypoxic chamber for the projects that we're doing is set to, I believe, 5% O2. And so it maintains an environment of 5% oxygen, 10% CO2, and then we'll, subtract that from 100. And then it's 85%, the balance is nitrogen.
Okay.
And so not year round, but at least project round until we wrap up this current set of projects that we have going on in there, it'll stay at that exact sort of composition.
And maybe just to compare to the composition of air in general, it's like mostly nitrogen, 20-ish% oxygen, and then a lot less of all the other weird gases.
Yeah. Exactly.
That we're not going to talk about
a bunch
today.
of N2, a smidge of O2, and then absolutely zilch of everything else.
Yes.
Yeah. What do you think the third most common gas in the atmosphere is? Because I think we're like nitrogen, oxygen, and I'm like, what's number 3?
It either has to be H2 or argon, maybe.
Argon? Okay. What's a...
I mean, carbon dioxide is probably also up there, but it has to be hydrogen, right? Doesn't that count as a gas?
I mean, if it was helium, everybody'd be so squeaky. Oh.
Man, should we look that up?
Yeah, let's look it up. My final vote, I'm going xenon.
I'm going argon, just so that we're like embarrass ourselves.
Yeah, that works.
lot.
I mean, that.
Only one of us is embarrassing
What?
themselves.
Do tell.
And for once, it's not me. What a day.
I was like, I pulled that one out of the bag. It was xenon. There's no way.
No, apparently, I mean, the AI overview
Oh, okay. Fair
says
enough.
argon. Wikipedia says argon.
Okay. So we are not chemists, folks. Atmosphere chemistry is not our area of expertise, dare I say.
That is true.
So we're going to...
and the NOAA, a trustworthy source, also says argon.
Okay.
And then the fourth, you want to guess on the fourth?
Oh, my. Well, if the third is argon, the fourth is going to have to be... I'll go hydrogen.
No, and hydrogen is the 1, 2, 3, 4, 5, 6, 7, 8, 9th.
Well, I jumped the gun on that one.
0.55 parts per million.
Whoa.
I mean, it's probably good, too, because isn't hydrogen pretty explosive? And hydrogen and oxygen might not be a good combination.
There's something to be said about that.
I didn't even think about that before. No, the fourth most common gas is carbon dioxide.
Ah, okay.
With 0.042%.
The fourth most common gas is 0.04%.
The third most
that
common.
Ain't much.
The third most common gas is argon with 0.93%.
Wow.
So it's really just nitrogen and oxygen.
Yeah.
But xenon is on there as the 12th most common gas with 0.09 parts per million.
Hey, look at that. Without the knowledge of looking at these results right now, how long do you think it would have taken the two of us to guess the top 10 gases?
I think we could have gotten like 5 to 6 out of the top 10 pretty quickly. And the rest, yeah, I don't think we would have gotten those. Because there is krypton in there.
Oh, okay.
There's neon in there.
Hey.
But yeah, hydrogen, helium, methane, carbon dioxide, argon, oxygen, nitrogen, those we would have guessed.
Oh, that is A-okay. Now we have went completely off the rails, but
Yes.
we now know something about the chemistry of the atmosphere. So,
And our listeners know too if we don't cut it out.
yeah, that's true. But we were talking about lab equipment. So small lab equipment. I think for me, this one is hard because I think we use a lot of the same types of small equipment that most labs use. We just might use them for slightly different applications. Or not even different applications. We're just, like say if we're making media, for instance, there's all of the typical, the serological pipettes and the Bunsen burners. And all of those things are the exact same. And we're making media just like anaerobic people would be making media. But having said that, I think the difference for us, you said it earlier, is like crimp sealed tubes. So we've got our bulge tubes, which are these fancy-ish glass tubes with a little lip on the top that we can put a rubber stopper in. Yeah, and the crimp seal on top. And so I think those would be different things that like other labs don't have / don't have a need to use. And then I think by default, like a decrimper, right? Because like once we've the experiment has ran its course. We're done with the particular tube. Well, you got to pop the seal off and then, clean the tube and sterilize it and prep it and get it stored away and all of those things. Mm-hmm. So, which takes a very short amount of time, right?
Yeah. No trouble at all.
Like
no blisters on your hand
per
from opening like a million tubes at once.
So there's no resentment.
No.
You harbor no ill will.
I harbor no ill will.
Okay. I'm not going to call you on that line.
Thank you.
Oh, but what else? Because I'm trying to think too. There's obviously the centrifuges and all of those things are the exact same.
Yeah.
I think
you use the same. Oh, sorry.
No, go ahead.
I was just about to say, in my anaerobic fungi world, we never pour plates because we always work in bulge tubes. But coming to this lab and seeing you work, that was really interesting to me to see that you just pour your plates like any other lab would pour a plate. You just mix the media in a shot bottle, pop it in the autoclave, and pour it into petri dishes. But then you just degas it. I don't know if you would call that, but you like pop it in the chamber for a while to like get rid of all the oxygen.
Yeah, that's true too. And so there are some companies out there that sell what are called PRAS media. So it's pre-reduced anoxic sterilized media. Mm-hmm. And oftentimes they come pre-packaged, and sometimes they'll be in like individual little packs. And then they'll have a little desiccant pack in there, and then, you'll just take it into the chamber, rip it open, and huzzah. You're like ready to rock and roll.
Nice.
So yeah, those are kind of fun. But I don't, maybe something that I would come up with are loops. So I tend to go through a lot of plastic loops just because the work that I do in the chamber, like we don't have access to a flame in there. Mm-hmm. And so by default, I can't use like a traditional metal loop, flame it, and go about my work in the chamber. So I go through a ton of plastic loops. And I'm sure that regular labs use plastic loops out on the benchtop too. But I think for me, I'd probably say that I go through a good volume of plastic loops. Mm-hmm. Yeah.
Because you can't use the other ones. I hope that regular labs that have the option to use the other ones do.
Yeah, we judge you.
Yes.
Harshly. Semi-harshly.
But yeah, I think the media making process, while it's sometimes like for you, sometimes it's very much like the aerobic media. And the only difference is you do your work in your chamber. The anaerobic fungi media, at least like liquid anoxic media that you use in bulge tubes, that process is a little bit different to the aerobic media production because you have to boil and flush it and whatnot, and it takes forever. But yeah, I think for media production, we just use what everybody else is using. We're not so different.
Yeah,
but we are.
All. Yeah. So that's the conclusion then, right? Is we use mostly the same stuff.
Yes.
And then we just have a couple extra toys and tools.
Yeah, we just need at least one of the large equipments and then two of the smaller equipments, and we're good to go.
Okay, so speaking of that large equipment, specifically like, say, the chamber and the gassing station, something that I wasn't really aware of until I became that glorious PhD student that had to work with it all the time is the labor of love that went into maintaining those type of things.
The labor of love.
Well, you can't just flat out say, I hate them. No, I mean, I guess you can, but, you just don't talk to them directly.
It's not polite in the Midwest to tell the truth.
Exactly. Yeah, it's like one of those bless your heart type situations. My Southerners in the U.S. will know all about bless your heart. Mm-hmm. But I think there's quite a bit of maintenance that comes along with, say, the chamber and gassing station. So I'm thinking for the chamber, obviously there's the changing of the tanks. We kind of talked about that earlier. We go through tanks, and that involves turning wrenches and lugging heavy tanks here and there and everywhere and getting them to a particular spot. And if you don't count the administrative stuff, of like ordering the new tanks and making sure they arrive on time and all of that. And then also, sadly, sometimes things leak, right? Like anybody that's worked around a glove box or an anaerobic chamber or some sort of an anoxic vessel like that, right? You've had to deal with with leaks, whether it's in the bag itself, in our case. Sometimes something will poke it, or it's just age. There could be leaks in the lines that are coming from the tank into the chamber. There could be leaks. I think for me, in my experience, it's most common to have the leaks in like the arm ports, just because most of your activity is essentially in those arm ports. They get like the most use and abuse. And then also the gloves. Like maybe you pierce it with something sharp and, you know, that kind of thing. So then you got to change the gloves. And so there's a bit of maintenance that comes up with that. And also things like inside of the chamber there should be some catalyst boxes where you've got, you know, a palladium catalyst that's setting on a fan box that's sucking in the atmosphere, that you then every so often you want to take those catalysts out and essentially reactivate them, stick them in a furnace, crank the heat up really high, and essentially re-energize those. Yeah. It's like that's something you have to do.
And the desiccant would go with that too.
Well, for those plebeians that have to put a desiccant
just
in
for us plebeians, for the ones that have the older anaerobic chamber.
Yes. You old folk out there, change that desiccant. Which I had to do that all the time in my PhD. But now I am so grateful that we have that dehumidifying attachment on the end of our koi chamber. Complete genius. I absolutely love that thing. But without going too far into the weeds there, like why desiccant, why dehumidifying attachment? Like what's up with the water that's in there? Well, that's essentially the fault of the catalyst because it's taking oxygen that's in the atmosphere, any sort of residual oxygen that sneaks in or whatever the case might be, smacks it together with 2 hydrogen, and then you get H2O. And so then that water vapor, if you don't have a desiccant, like it's going to stick onto your metallic surfaces and you'll see rust in chambers and things of that sort. So if you're not keeping up with those desiccant changes, or you're not super fortunate like I am to have that dehumidifying attachment that's just constantly sucking out water vapor, you can really have some like big time corrosion problems going on. Yeah.
And also, I mean, if you have a closed system that you handle liquids in, you will always get water vapor. So
yeah, no, exactly.
Yeah. It's really important, the desiccant and the catalyst are like, I think, the most important things in the chamber itself, other than the chamber being airtight.
Oh, yes.
Yeah.
Yeah, I can't tell you how many weeks sometimes I would spend trying to fix and patch holes in the chamber that I did my PhD in. And it would be like, blow the bag up, try to find the hole, can't find it, bag goes down. Blow the bag back up, try to find the thing, it goes back down. And so all over that bag, just what the convention was in my old lab was you would put a little Sharpie circle around like wherever the hole was. And so you would kind of know like, okay, this is where that hole was that I patched, you know, at X time. And then, you know, you patch it appropriately and move on with your life and hope to the anaerobic gods that it doesn't happen again.
And hope that none of those holes are in like your line of sight.
Yes, that too. Oh, and even cleaning in the chamber, right?
Oh, yeah.
Like maintenance like that.
Yes.
So that's something that we do slightly differently up here because it's predominantly just ethanol based.
Oh, yeah.
Right, good old 70% ETOH. Shout out big ethanol. In my old lab, we used to use one normal HCl, and that was mainly just to get rid of like endospores. That was really the only difference between the two. I guess the caveat there is that sort of as that one normal HCl, it's kind of sort of implied, but it's a bit more aggressive than 70% ethanol. And so it can certainly tarnish things and, you know, make it less shiny and less pretty and all of that.
Yeah.
All of that good stuff.
Oh, but the gloves would like be resistant to it, right? Because those thick rubber gloves that go into chamber would be resistant to that.
Yeah. I suppose you know.
That's neat. I've never heard of that. Nice.
Got a hole in your glove.
Yeah, that might not be good.
Yeah, don't put that on your hands, kids. Don't do that. Don't try this at home. Yeah. I'm trying to think. I don't think there's really anything. I mean, I'm sure there are other little nuances to the chamber life, but
yeah, just maintaining order in the chamber, and then
I think we said this a couple podcast episodes back, but it's mitigating chaos.
Yes,
that's essentially what we're doing here.
Yeah,
we're the Chaos Crusaders.
And everything you need in the chamber needs to fit in the airlock. Like you can't have anything bigger than the airlock going into the chamber.
That's true, but you can. You just have to then deconstruct your chamber.
Oh, yeah.
Right? You take in the case of the koi, It's got a big, circle port on one side that you can then take off, stick equipment in, reseal it, presuming you do that correctly. Wink, wink. And and then blow your chamber back up and do.
Yeah. If you want to make your life harder, you could do that. Or you could just try to not need anything bigger than whatever goes into the airlock.
Yes, Sam is a big fan. I'm talking about myself in the third person here. Dr. Ambassador Sam is a big fan of the KISS method, the keep it simple, stupid method. So yes. I'm all about, not overcomplicating things.
I completely subscribe to that.
Yes. But that's the chamber maintenance, I think. What about the gassing station, Julia? What do you find yourself doing to maintain that sucker?
I think it's a little bit easier also because you have the full range of motion in your hands without the huge gloves of the chamber. Maintaining a gassing station, I think, is way easier than maintaining an anaerobic chamber because what you really want to take care of is just the tubes coming out of the station. That's, I think, the main thing. Of course, all the tubing from the gas tanks into the station, into the tubes need to be airtight and sealed, of course. So that needs to be set up correctly. But other than that, it's just maintaining those little tubes that come out of the gassing stations because they tend to go brittle over time. They might need to be exchanged. And I think that's the main thing because otherwise, of course, you have to exchange the gas tanks when they're empty. But
yeah, I think I'm totally in agreement with you there. Yeah, aside from obviously the potential leaks and the changing of the tanks and all of that stuff, which is more or less the same as it is with the chamber every now and then.
And I mean, we don't do anything weird with our gassing station. We don't dunk the tubes into anything. So the tubes are fairly clean most of the time. I mean, you wipe them down with ethanol if you put something on there. So it's really just exchanging the tubes, which is, to be fair, it's a tedious process. You have to get open those little screws and everything, and then you have to put the tube into it again, which is insanely infuriating sometimes. Because it's a very tight fit, of course, because you need it to be sealed.
Yeah.
But yeah, it's fairly easy to maintain a gassing station. I think once you have it, I think building it is the main part.
Yeah, I would totally agree. I think the building of one would be tough.
Yes.
That's on my postdoctoral bucket list.
To build a new one?
Is to build one, from scratch. But please don't hold me to that, internet.
Listeners.
Yeah, don't.
Do you want to see
Sam
build a gassing station? We could do a fast-forward version of
that time lapse would be 4 days long. It'd just be me going, oh. Yeah. Oh, that's something that I meant to ask you. How do we detect if there's any leaks?
Oh.
If we presume that there is a leak somewhere, be it on a piece of tubing or if it's on the chamber or wherever it might be, what's your go-to findy hole in the thingy?
I mean, if you've ever had a bike, you know how to find a leak in something that shouldn't have a leak because soapy water is an option to just put the liquid or the soapy water onto the tube, for example, the tubing, and then blow gas through it, and wherever it bubbles, that's where the leak is.
Okay.
If that works. And there's of course, we're in the lab, we're professionals. There's no soapy water involved. There's this special gas leak finding liquid, which I guess is just soapy water.
Yeah. So in a pinch, some dish soap and some water.
Yes.
And you'll be okay.
Yeah.
Yeah.
I think so. Although on the gas, on the anaerobic chamber, which is huge, that is more of a troublesome business still.
Yeah. I will say that I sometimes enjoy using the leak detector from time to time. So if there's an issue with the chamber, one of the things that they ask you to do is to soak a rag or a towel or something like that in a particular hydrocarbon, something of that sort, stick it into your chamber, let it set for a few minutes, and then you turn on this little, I joke, I call it the anaerobic Geiger counter. I turn it on, it's click, 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 click. It's just going crazy, and you think that the world's going to, well, maybe it will blow up, who knows at this point, but in the anaerobic world here, it's got that Geiger counter going, and it's sort of acclimating, and then after it, you follow that protocol and you let it acclimate for so many minutes or whatever the instructions say. Then you take it and you kind of just go around the tank or not even the tank, but the chamber. And so I'm in there, and I feel like a proper scientist from like the 80s doing something very important, you know?
Does it come with headphones?
No, it sadly does
Oh,
not.
no.
No, everybody hears the beep. And then essentially you dial the beep down before you start this process after so many minutes of like warming up. And then it'll just start out slow, and it's like beep, 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 and then you just keep going around. And as you get closer to like the leak, the idea at least is that it then starts going faster, and then it's like beep, 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 and you're like, oh no, it's another issue with my glove, like wah. And it's not foolproof, but I think it can help at times, especially if you have no idea like where to start with your leak detection.
Oh yeah, I can imagine that this is easier to use on an anaerobic chamber than dousing the whole chamber in like soapy water. Even though that might also be a good idea from time to time.
Just give it a bath. Yeah.
Nice.
So maybe there's another piece of equipment. We got the little leak detector Geiger counter machine. Maybe I'll turn that on sometime and we'll do a little video.
Sam's favorite lab toy.
No. not even close.
Do you want to elaborate on that? What's your favorite lab toy?
I mean, I enjoy a good multi-channel pipette.
Oh.
I keep going back to that multi-channel. It's like I can't, or a good serological pipette, hands down. I think those are probably like things that I use so often, but also things that I genuinely enjoy using. So probably for me, it's one of those two. What about you? Do you have a favorite laboratory piece of equipment?
I mean, the microscope is like my favorite.
The microscope.
The microscope. I don't care which microscope. No, I do care. I was care.
Say, we've already figured this out. We've sussed this out, folks.
I do care. But like the microscopes we have, I could sit there all day.
Yeah.
Yeah.
Okay. Well, perhaps we need a microscopy episode in the near future.
Perhaps we do.
Wink, wink. Okay. Let's see. Back to planet Earth.
Yes.
Allegedly.
I was wondering because I remember back in was it the first episode or the second episode? I can't remember. But you were talking about a piece of equipment that you always keep in your car. And you've not mentioned that piece of equipment yet.
Oh, the 50 mil conical tube.
No, not that one.
that might be my favorite. I digress. No, I know. I presume you're talking about like an anaerobic jar.
Yes.
Yeah. No, that's fair. That's fair. So yeah, that's probably something that our lab has that that anaerobic labs in general have that most regular aerobic folks probably don't have is the need to have these little portable anoxic systems. And so an anaerobic jar, I would pitch that to you as it's like a portable anaerobic box. They come in different shapes and sizes. Some of them are like tubes, and they're made out of different materials. Some are metal, some are different plastics, and then there's some that are more of like a rectangle or a box, and then some that are just like a glorified Ziploc bag. This is a really fancy type of plastic bag situation. So there's all sorts of what I would collectively deem like anaerobic jars. And essentially those, what we'll do is we'll put samples in there, or we'll put, say, in my case, plates or something like that, something that needs to be incubated in an anoxic environment. And then I will toss into that anaerobic jar either what would be called like a gas pack, which would be something that you would essentially rip open and stick it in the jar and then close it. And then what'll happen is any sort of oxygen that either gets into the jar that was in there to begin with, it starts converting it to water vapor, and it fills the atmosphere with a particular composition of gases that's devoid of oxygen. Or some anaerobic jars you can actually put your own gas into. And so those ones, most of the time on the top of them, they will have a little valve stem, similar to like if you wanted to put air in your air, put air in your tire. And like during my PhD, it was literally like a glorified bicycle pump that we would have a line off of the gassing station, and it would be like, okay, we're going to douse this one with an anaerobic mix, or we're going to douse it with N2 or something like that. And then we'd push the atmosphere in through the bike pump, through the valve stem, into the jar to a particular pressure. And, but yeah, so those anaerobic jars, again, you can buy little catalysts for them, or you can put your own gas blend in there if you want to.
Nice.
Assuming you've got that kind of setup. Yeah.
That's pretty cool.
Yeah, they are pretty cool. And it's handy too, because oftentimes you might not be able to incubate something in the chamber, right? So if you're one of those lucky type of individuals that have a chamber with an incubator, whoop, whoop, obviously you can use the chamber incubator, but you might need to do something outside the bounds of its temperature capabilities. Otherwise, you're limited to like the temperature range of the incubator as well as room temperature.
Yeah.
But I might want to do, these anaerobic cultivation efforts at like 4 degrees or at 65 degrees or something wild like
Crazy.
that. Yeah, something just bananas. And so those jars essentially make that possible.
Nice.
Yeah.
Yeah, that's like, I don't know what your first memory of anaerobic microbiology is, but I think my first touch with anaerobic microbiology was in the Intro to Microbiology class during my undergrads. And I specifically remember our professor telling us, he was a aerobic microbiologist, but he mentioned that there's anaerobic organisms out there that might be facultative or strictly anaerobic, and there's different equipment to deal with. And I really remember those pictures that he had up on his slides. One being one of those glass tower jars where you can put the plates in and then close them up and fill them with gas, and that thing you could put in the incubator. And the other image was just a comic. You would see a little tube of a microbe swimming around in media. And then he would describe that some, with anaerobic organisms, sometimes you can just put a layer of oil on top of the liquid to seal off any other incoming oxygen. And then there was this little microbe comic that screamed, "But I'm not anaerobic."
Oh, that's good.
That one stuck with me.
That's good. So kudos to that.
Shout out Professor Blasey.
Blazy. That's a cool
Yeah.
last name. I'm assuming that's a last name, Professor
That is
Blazy.
his last name. I just can't remember his first name now. He was hilarious. I really enjoyed the Intro to Micro classes. He was also really, really smart, and it was sometimes hard to follow because he would be going through it quite quickly, and he had high standards and stuff, but I just loved listening to him. He was amazing.
Those people with high standards.
Yeah.
Yikes. Wow. I'm trying to think of my first encounter with anaerobes. It had to have been in an undergraduate micro course. And the only thing that's really coming to mind is we did these, what were they? They were called fluid thioglycolate broths, these FTG broths.
Wow.
Were
That's a
essentially,
word. We're throwing out other big words now.
I know. Google that one, kids. But it's the stereotypical image that you see in the micro textbooks where it's a tube and then it's aerobes grow on the top, anaerobes on the bottom, facultatives throughout. But it's sort of that imagery because the way that the tube is designed is it essentially creates an oxygen gradient throughout the tube. And then what I find quite cool about that tube specifically, those FTG tubes, is the way that they're inoculated and set up and constructed is that you can even see the gradient because there's rizazerin in there. And so it's pink at the top, not pink at the bottom kind of situation. But anywho, so that's probably my first memory of anaerobic microbiology is putting drops of culture in that and then having to wait till next week to figure out where the cloud was going to be.
Nice.
Yeah.
But that brings us directly into my next question because you mentioned rizazerin.
Oh,
which is also one of those big words that is rather easy to say, but not many people probably know what it is.
Yeah. I'm not even sure I know what it is.
Do you care to expand on this one? Or do you care to expand on what we're using it for?
On rizazerin? Well, I think we've joked a bit about rizazerin in the past, but rizazerin, for all intents and purposes, is a redox indicator. And so why we use rizazerin is we essentially use it to gauge whether or not there is oxygen in our media. And there are other redox indicators out there, things like methylene blue, But what these do is essentially in the presence of oxygen, they're a particular color. And when there is no oxygen, they're a different color, more or less. And so with Rizazarin, there's really 3 colors. So there's colorless, there's blue, and there's pink.
Keep going.
Oh no. Feel free to jump in.
No, it's just like those are the base colors, but it depends on the media that you have, what color it is.
Yeah, that's true. I guess I should have laughed. So we'll start with blue and pink. Let's start there. Yes. Colorless we'll circle back to. So blue and pink both mean you got oxygen in there. Mm-hmm. Blue is like the most oxygen, which is usually how Rizazarin starts. Oftentimes, I think if you have like a stock solution of Rizazarin at such and such concentration, it's probably blue.
It's this vibrant, really pretty
It
blue.
is a very pretty color.
Yes.
I was actually thinking about changing my PowerPoint slide default color to that blue.
Oh, to Rizazarin blue.
I genuinely was.
Yeah.
And I might just do that. And then pink, it means that it's present, but in a little bit lesser of an amount, but it's still enough to, take out your obligate anaerobes. Mm-hmm. And so for us, sometimes when we're manipulating tubes, especially outside of the chamber, as you're going in and out with syringes or you have different stock solutions that you have to put into your media, sometimes inevitably there's oxygen that sneaks in. Mm-hmm. And then you'll see a tube turn a little bit pink, and you go, oh, what a waste of my life. Here we go again.
Although sometimes they settle back down, because Rizazarin is actually, it doesn't detect oxygen per se, but it's like a redox indicator. It turns pink because there's some oxidation reaction going on, maybe not including oxygen or molecular oxygen. And then it settles back down to whatever color it is that you had before, while it was still anoxic, and your life is good again.
Yeah, I was going to say that you get right back into the good life. And so I said colorless earlier, perhaps carelessly. But what I meant by that is whatever the base color of your media is. So if your media is quite literally as clear as water, then it would turn colorless. And so sometimes media is a little bit yellow, sometimes it's a little bit brown, it might have a particular color situation going on, but then you add the Rizazarin to it and you start instantly seeing the color change a little bit, presuming it's not too dark, which is, I think, the thing that we chatted about previously on here is that it's, at least in my opinion, in my experience, Rizazarin is not as effective in darker colored media. Mm-hmm. Yeah. Because I remember one of the first anaerobic things that I was doing in my PhD is we had gotten this culture of Blautia, which is just this obligately anaerobic genus of bacteria that's commonly found in the gut. And I'd gotten a particular type strain from a culture collection. We were trying to grow it, and it was grow it in PYG. That was the media of choice for the culture collection when they gave it to us. And so I made up this PYG, and it had all of this stuff in it, and it was super dark. And then they're like, you got to add Rizazarin, Sam. And I'm like, but when I add it, it's is it slightly darker? I don't know. So I'm not a giant fan of putting Rizazarin in very darkly colored media. I would rather just not have it.
Yeah, I would totally agree. In very dark media, there's no point really in adding Rizazarin.
How pessimistic of us. I will say, though, sometimes at the MCCF, so I suppose this is where I say the Microbiomics and Culturomics core facility here
For
at
those
OSU.
of you who don't know.
Yeah, if you haven't already heard me say that 17 times, not that I'm keeping track. We'll have a client that they need us to help them make media. Mm-hmm. And I'll give them the media, and then in addition to giving them, say they need 20 tubes of media, because I'm nice and so loving, I'll give them 25.
Wow.
And, I'm a sweet guy. And then I will give them an extra tube that has oxygen in said media. Mm-hmm. And then I'm like, if it looks like this one on the upper right corner, don't use it, because your babies are going to die. But yeah, I usually try to do that, because I think especially to the untrained eye, it can be subtle.
Yes.
Yeah, very subtle.
That's also what I do with my students and all. I try to keep oxygenated tubes at hand and show them hey, this is what it looks like if it happens.
Yeah, no, that sounds good. I know when I was getting heavy into the anaerobic culturing there during my PhD, one of the exercises that I was told to sort of go through with myself is essentially I made up some anoxic water in a couple of different bulge tubes, and then I would just, at the gassing station, take and transfer water from like one tube into the other. And then it's like, you know, you watch them sort of turn back pink, and you're like, oh, okay, I'm still horrible at this. 3 weeks later, I'm still horrible at this. Eventually, it goes away.
Yeah.
But yeah, that was sort of one of the exercises that I undertook to get accustomed to not screwing up my anaerobic tubes and thingies. Yeah.
I think that's also a fantastic point, comparing anaerobic work to aerobic work again. I mean, in any kind of lab work, you have to learn the handling. Like there's a learning curve to the handling steps and how to efficiently hold your hands and use your fingers to like do any of the steps to not contaminate something, for example.
Yeah.
But I feel like there's a bit of a longer learning curve to anaerobic work because it just takes, it's just so weird. And I don't know why, but it just takes a little bit longer than you would take to learn like aerobic work. And also, I mean, aerobic work during your studies, well, like, at any point, like probably in your undergrads, you have like a microbiology lab.
Right. I was going to say you're exposed to that.
Yeah.
Yeah. You're not really exposed to like making anoxic media in an introductory micro class. Or even in my case in grad school, there was never like a anaerobic microbiology 101. You know, like, hey, this is how we make media. And this is, I don't know, the parts of your gassing station. Get to know it.
Yeah.
It was more like jumped off a cruise ship into the Atlantic and try to find an island and don't get eaten by sharks whilst swimming. And oh, there's a hurricane coming, but don't worry about that, Sam. It's fine. And you're in the Bermuda Triangle. I take that. I take that back. Maybe it wasn't that bad. Or was it?
Yeah.
Am I coping?
Well, I mean, you made it through it, so.
Allegedly.
But it was the same for me. I was never exposed to anaerobic work other than that nice little comic from Professor Blasey. So I had also no idea when I started anaerobic work in my grad years. And then I immediately started with anaerobic fungi. That's like, hey, you've never worked with a difficult organism and you've never worked with an anaerobic organism. Let's go.
Yeah, let's go. Go for some of the really, really tough ones.
Yeah. Let's see how resilient you really are.
Yeah. Now, speaking of resiliency, horrible segue here. We just talked about the making of the media, the tubes, they're turning different colors, or perhaps they're not. But there are some other tips and tricks that we employ to get the oxygen out of our media. And in one of our previous episodes, we sort of talked about the importance of boiling the media to help get rid of those dissolved oxygens that are hanging out in the water. But there was something that we didn't really touch on that we also do to our media most of the time, and that's adding in an oxygen scrubbing type of molecule.
Oh,
so we use them.
We're cleaning the oxygen. We scrub it clean.
Yeah, I mean, why not? Oxygen, you dirty molecule, you O2.
Hey, that's the wrong
everything.
podcast here. That's not the kind of podcast we are.
Yeah, we're not hating on oxygen. Well, I mean, we are. But
We are.
But it kills my bacteria. You scoundrel. There we go. Oxygen's a scoundrel. But what oxygen scrubbing molecule? I mean, there's several of them out there, but the main one that we use in the lab, Julia, what would you say that is?
It's our Bye-Bye O2 powder. If you've ever seen our We Are Postdoc clip.
Yes.
And that's cysteine.
Yeah, the Bye-Bye O2. Yeah, good old cysteine. But we add this cysteine to our media to essentially lower that oxidation reduction potential. And it's almost like these layers of built-in redundancies, right? So we can be overly cautious and so we can essentially make absolutely sure that there is no oxygen in our media. So we're boiling the media out, that gets rid of the O2. Mm-hmm. We're flushing with anoxic gas. We got an anoxic headspace. But at the same time, we're going to pump in cysteine and, any sort of residual oxygen that's more or less going to take care of. And there are certain microbes that can utilize those as sort of a source of sulfur, source of nitrogen. And then on the flip side of that, there's also depending on the concentration of cysteine that you've spiked into your media, it can also be inhibitory. And so it's kind of one of those Goldilocks type situations where you don't want there to be too little or too much. You want there to be sort of just the right amount of cysteine in your media.
Yes.
Yeah. So I said earlier that I don't usually put Rizazirin in, darker colored media. Like I feel very, very confident when I make anoxic media, especially now, where I'm like, okay, I've boiled it. I've done all the things. I've got, X concentration of cysteine going in. It's been autoclaved. It's got all the nitrogen or whatever in it. I'm like rather confident at that point that I'm like, don't really need that Rizazirin.
Sam's feeling frisky.
And then my stuff dies. And I go, why, God, why? So
yeah, sometimes you just can't figure out why it dies.
Yeah, so inoculate more tubes than you need, kids. That's the moral of that story. One of them doesn't work.
And add your damn Rizazirin, even if you're confident that you don't need it.
Yeah, do as we say, not as we do. There we go. That. Now, we've talked a lot about making media, and that's sort of where this conversation has went today. And I wanted to ask your opinion on something, Julia, and that's sort of there's two different broad categories of media. There's defined and then there's undefined. A defined media, for those of you out there that aren't super familiar with this idea, it would mean that you would know exactly how many milligrams of each vitamin, of biotin, and this and the other thing, and how much glucose and how much of this. Like you would know exactly the right amounts of every single thing that's in that tube. Whereas undefined, it's like, toss in some yeast extract. We don't really know what that is, but it's good stuff. Yes. Toss in some rumen fluid. Toss in some, ground beef, whatever it is. Get it going.
What are you guys doing over there?
That's chopped meat media. That is gold stuff to us. Ugh. I'll show you some when we get back to the lab. It's got little pink pieces of like 80-20 ground beef. Please don't listen to this podcast before lunch, people. But for real,
those bacteria people.
That's a very, very common media, and we'll be extra nice. We'll spike a little glucose in there. They'll love it.
Nice. That's the American diet right there: beef and glucose.
I mean, if you're a Clostridium and you want to grow, I got you. So maybe you could try your AGF and some chopped meat with carbohydrate media. We'll see what they do.
I don't think they would like the high concentration of protein in there.
Yeah. Well, so clearly AGF aren't American.
AGF are vegan. They're like...
Oh, there we go.
Because they eat grass.
That's their problem. No, we love our veganistic AGF.
Do you even know what the word vegan means?
Tell me.
I don't think that most of the Midwest or the South or whatever you call Oklahoma even knows what vegan is.
Yeah.
Like anything that is taken from an animal without that animal's consent is not vegan.
So, on that note, I have to say your AGF are not vegan.
I was about to say, like, are researchers vegan? Like, is that what we're doing? Vegan? I don't think so. I think
you got that rumen fluid in there.
Oh,
that bovine rumen fluid.
Yes, of course. Yeah. We do, but we do have defined media. So if they're swimming around in the defined media, that might be vegan.
Okay.
Although we still keep like them caged in tubes and they're not allowed to like roam freely. So I don't know if vegan or not is the debate we have to have.
I mean, you're not wrong there.
Okay, we went off the rails really quickly.
Yes, defined versus undefined media. So, do you find yourself using more defined or more undefined media?
That is actually a tricky question. One copy of our culture collection is on rumen fluid cellobios media. The other copy is on rumen fluid free media. So I would say it's 50-50.
Yeah, I'm trying to think too, and I think for me, I'm gonna hang on the fence here and say that it depends on the project. In general, blanket statement, I think I use more undefined than I use defined, because a very common couple of media in the anaerobic microbiology are like brain heart infusion.
Oh my God.
Yeah.
Like beef brain heart. What the F?
Hey. So that's big. We obviously spike things with rumen fluid, and then I also add in defibrinated sheep's blood to auger plates.
From a future attorney, I never went into that lab. I have no idea what they're doing over there.
Hey, we buy this from approved scientific channels here. We're not like going out back and vampiring the dolly.
Do you have to turn on a VPN service before you order that sheep's blood?
No, I usually send you a link and say, hey, buy this. But
again, from a future attorneys, no, I didn't.
Oh yeah, true. Well, yeah, Ty did that. We'll say he did it.
Nice.
Yeah, that works for me.
Yeah.
Yeah, so I think for the most part, it's probably more undefined than it is defined. Although there are times when I have to make defined media, and then I will do that. And so an example of that I remember is like having to get in particular proportions of like volatile fatty acids. And I had to make these specific cocktails to then put in a pretty minimalistic type media to grow things. And that was like, it's always annoying when you have to do proper science. And you know, try to get everything dialed in exactly right with concentrations and proportions and all that stuff. And yeah, but I think for the most part, it's probably, I'm going to say it's an 85-15 split for me.
Nice.
Undefined versus defined.
And I just have to redo my answer because I remembered that in our defined media, there's yeast extract in there. So it's also undefined. So
that's fine. We'll still call it defined. Semi-defined. There we go. And I will say, maybe I'll comment more, or I will lament my sorrows more in the future about these defined media as I am working through characterizing this one novel that we have in the lab. I don't think I've talked about it yet on here, but I suppose I'll just launch it out into the world. So we have a novel isolate belonging to the negativicutes.
Negativicutes.
Yeah, negativicutes are, a class within formerly firmicutes, but the real ones out there, Bacilliota, what's up? I mean, I have nothing against firmicutes. I grew up on firmicutes, but...
Okay, so the negativicutes are a class within the firmicutes.
Yeah, and so we believe currently that we have a representative isolate of a novel order within this class.
What?
Yes, which complicates my life for the next several years probably as we work towards doing a really thorough characterization of this isolate. And so things like all the substrate utilizations, all the biochemical reactions, electron acceptors, donors, like stuff like that. It's going to be, painful that's the right word. It's going to be painful.
Nice.
So as I continue to work that culture into, submission and get it to grow in a very minimal media, I will continue to lament my sorrows.
Okay.
Yeah.
So you're lamenting about defined media. I will lament about autoclaves.
Yeah. Oh, back to the autoclaves. All right, folks.
I was just thinking, like, now you have your topic that you can lament about. All
time since last autoclave rant, 33 minutes. Do tell.
No, I just think it's nice that we both have something that we can continuously lament about.
Yeah.
So stay tuned for this very positive, optimistic podcast.
Yeah. Yeah, I can't. So I'm trying to think of all the weird stuff that I've used over the last few years. So you got, rumen fluid, obviously. We chat about that quite a bit. Thank you so much for preparing a large batch of that the other day. I spent a lot of time at my desk outside the lab.
I was wondering why I didn't see you in the lab.
That was planned. Then like defibrinated blood, laked blood, horse serum. There's all sorts. Pig mucin. The list goes on. And, I guess now you know about the ground beef. So
yeah, I'm gonna smile and nod, and then the moment that door to the chamber opens, I flee.
Yes. So next time we're having a lab grill out, and I'm over there.
No,
I'm getting the burgers going on the grill.
You are forbidden for going next to a grill or bringing any food to lab lunch.
Oh, there we go.
Because the next lab lunch is also our weird lunch, right?
Oh, I know.
So I'm like, we should not have gone with that idea, because Sam is definitely going to bring his beef patties doused in sheep's blood.
Yeah, well, I'm not gonna be that. I'm not gonna be that mean about it. But maybe I could do like a ground beef in a glass, like a little shot of something. That's what I'll do. I'll do like ground beef and like maple syrup or something. You people want to play? Let's play.
And I'm getting roasted for my peanut butter pickle sandwich.
Yes, you are. That's a disgrace of a sandwich. Oh, that brings me to something that we should probably talk about. This is my lab yap. This is my soapbox, and I'm going to climb on top of it.
Lay it on us, Sam.
Like King Kong at the top of the Empire State Building. Holding that lady in his hand. I'm holding a box of pipette tips from you psychopaths that take them out of the middle. Okay. All right. For those of you that are not intimately aware about how a pipette tip box looks, generally there be 96 of them in there. Okay. You got 8 rows and 12 columns. All right. People that have an intact conscience and have strong moral fiber, not even strong, they just have a moral compass. It may be pointing in the wrong direction, but they still should, to me, intuitively think, hey, I'm going to start in one of the corners, be it the top right, the bottom left. Just start in a corner. I won't judge you if you start in the bottom left. It's better than starting in the middle. But you start in one of the top corners and you work your way down. If you work your way across, it's okay. It's fine. But my God, those people that take like 3 of them out of the middle, and then they take one from the side and then one from the other side. Mm-hmm. What is the deal? I don't want to say that it angers me. Maybe it angers me. I think we're past.
It doesn't anger Sam at all.
He's just over here.
Chill.
Blood pressure is spiked, beet red in the face, trying to. Okay, so what? I'm angry. I'm angry, and I hate your guts. Yeah.
Yep.
What?
Yeah, I completely agree. I 100% agree. And I think psychologists that are doing research on like the dark triad, like psychopaths and sociopaths and all, in your questionnaires, when you try to figure out who is part of this dark triad, just give them a pipette tip box and tell them to take one tip out of it. If they go for the middle, psychopath, right there.
Yeah.
You don't need 50 questions to figure it out.
We just end it right there. That was lovely. I completely agree. And it's easy for me to sit here now, having done this, for the better part of a decade, and being like, obviously, do not do that, child. But I don't think there was ever a time in my life where I just went willy-nilly to the middle of the pipette tip box and started whack-a-moling them. Like, what the heck?
Of course not. Of course that you didn't. And before recording this episode that we recorded today, I would have said, of course not, Sam. You're normal. Now with your beef extract and your blood in your media, normal might be a stretch. But of course you didn't do that.
Thank you. So we thank those of you that have made it through this cumbersome episode, through all of the bloods and the rumen fluids and the redox and how to detect things with a Geiger counter. We appreciate you.
I feel like, Sam, in this episode, we've gotten to know you a bit more even than in our this or that episode where you revealed weird truths about yourself, like the 200 microliter pipette. But this episode was really, we got to see the real Sam.
What? I let the real one out of the cage?
Yeah, I think it slipped there for a moment.
Okay. Oh, no. Down goes viewership.
No. I think the viewers and I appreciate that. It was very fun getting to know you and talking about all of our equipment.
Yeah. I think if anything, something that we hope to do here is to be not blatant liars to you all, but to, tell.
Where are you going?
To tell, I'm going to spin this, I think. I'm going to attempt to. But we're trying to provide you all with information that we see as valuable as it pertains to anaerobic microbiology, and to also be real about some of the ups and the downs of, whether it's life in the lab, characterizing things, writing manuscripts, dealing with reviewer 2, anything that you might have to deal with. We're going to try our best to be honest and provide value in some sort of capacity.
Nice.
So thank you all for hanging in there.
Yeah.
That's what I'm trying to say. Thank you.
Yeah.
We appreciate you.
We do. We do. I didn't think you were going this way, but I completely agree.
Oh,
thank you.
okay.
Thank you all for listening.
Yeah.
If you have any questions for us, like if you're listening to us and you think, what the hell is the thing? Or what are they talking about? Or why the hell are they using beef junks in their media?
Yeah.
Any sort of questions, hit us up on our what are we calling it these days? Internet presence.
There we go.
LinkedIn.
Hit us up on the internet, kids. Yeah, we're all over the place. We've got LinkedIn, Blue Sky, YouTube, Spotify, Apple Podcasts. We're all over the place.
Yeah.
Yeah.
You could just drop us a message there with your questions, and we will try to answer them in one of our next episodes.
Yep, that's true.
Nice.
That's true.
Well,
okay.
Shall we wrap it up for today?
I
we
think
are getting
should
efficient.
Wrap it up and put a bow on it. Stick it in the autoclave.
Okay.
Okay. All right. Well,
I'm scared to press the stop recording button because I feel like there's more in Sam.
we can. To be. Stay tuned, folks.